Ladies and gentlemen, welcome to It's Rainmaking Time. This is Kim Greenhouse. It gives me great pleasure to welcome Dr. Bruce Lipton, a cell biologist who has discovered a major breakthrough having to do with cells, consciousness, and the biology of belief. In fact, he is the author of The Biology of Belief, Unleashing the Power of Consciousness, Matter and Miracles, and co-wrote his new book with Steve Behrman called Spontaneous Evolution, Our Positive Future and a Way to Get There from Here. Bruce Lipton is a pioneer, and what we are finding out through his efforts is that the mind controls the body through placebo and through other means, that DNA does not control our body and our health. This is a mind-blowing breakthrough that was represented in The Biology of Belief. Spontaneous evolution takes us into the realm of the morphogenic field, into spontaneous remission and healing, into how the mind controls the immune system and beliefs control biology. We talk about epigenetics and how 70% of what we think is really negative and redundant. But there's so much more. Ladies and gentlemen, welcome pioneer, cell biologist, author, and speaker, Dr. Bruce Lipton. Welcome to It's Rainmaking Time.
Kim, thank you so very, very much, and thank all the people out there, the inquiring, inquisitive minds that are open to hear some exciting and very empowering information. Thank you.
For those people who have not read your books yet and have not heard about you, I want to do a quick frame of reference about the cell membrane and why the cell membrane is important and related to the mind and the body.
Okay, well, let's first, I'll talk about the very simple experiment that just blew my mind. And I was a professor in a medical school, and I was cloning stem cells. Actually, I started cloning stem cells in 1967, so that's 45 years ago I was working on stem cells. And a lot of people think they're relatively new in our world, and the fact is, no, I was back there with some pioneers in the original days with stem cells. And here's what I did, and I'll explain it, and then hopefully the experiment will go, oh my God, I hope you say that. And it works like this. I would isolate one stem cell. A stem cell is a multi-potential cell. It's like an embryonic cell in your body. Your body is filled with these stem cells for a very simple reason, is that every day you lose hundreds of billions of your cells just to aging and damage. And so you have to replace hundreds of billions of cells a day. And the question is, well, where do you get them from? The answer is stem cells. So if you're alive, you have lots of stem cells, because otherwise, without stem cells, you would die out very quickly if you couldn't replace the bad, damaged cells. So we all have stem cells, multi-potential, the equivalent of embryonic cells. I put one cell in a petri dish. It divides every 10 or 12 hours, so first one cell, then two, and then 4 cells, 8, 16 cells, 32 cells, etc. Couple weeks, I've got thousands of cells in the petri dish. But here's the fact. They all came from the same parent cell, so by definition, they're genetically identical. That's the clone. They're genetically identical cells, thousands of them. Well, that's not the experiment. This is the experiment. I take the cells out of that big one dish and split them up into 3 smaller dishes. And I feed each one a culture medium. The culture medium is the environment. Cells are like fish. You have to put them in an aquarium, and you have to, put in the stuff that they live off of in that aquarium. So I put culture medium in the dishes, and here's what I did. I had 3 dishes with genetically identical cells, but I added culture medium with slightly different chemical composition. So the environment was different in each of the 3 dishes. In one dish, the cells form muscle. The second dish, the cells form bone. And in the third dish, the cells form fat cells. And then you're left with a very interesting question. What controls the fate of the cells? Well, the first thing is this. They were all genetically identical, so you can't say the genes controlled this. The fact they didn't. It was the environment. It was the composition of that culture medium. As I changed the chemistry of the culture medium, I changed the genetics and the fate of the cells. And it's like, oh my God, the cell is not being controlled by the genes. It's controlled by the response to the environment. I go, okay, now, big jump, but so profoundly important. Oh, before I say that, I love this. Take my dish of stem cells from a healthy environment and put it into a less than optimum environment, and the cells start to get sick and they begin to die and the culture crashes. if you were looking at this as a medical doctor and you look at my 6 cells in the dish, you say, boy, you should give them some drugs. And I go, no, absolutely not. You want the cells to get healthy? There's one simple way, and that is take the dish from the bad environment, put it back in the good environment, and the cells will instantaneously get well. The point is, the state of the cell is a reflection of the environment, okay? Now, cells in a plastic dish, interesting, but let me talk to us about humans. When you look in the mirror and see yourself, Kim, you see one individual looking back. You say, yeah, that's one human being. I go, well, there's a misperception because we're not a one thing. Because by definition, a human is created from 50 trillion cells. So by definition, a human is a community of cells. In fact, by definition, a human is a skin-covered petri dish. Because under your skin, you got 50 trillion cells. And then I say, yeah, and you got growth medium. The growth medium is called blood, okay? So here's the simple point. Whether the cells are in a skin-covered dish or whether the cells are in a plastic dish, they still respond exactly the same to the environmental information. In the plastic dish, we call it growth medium. In the skin-covered dish, we call it blood. I say, yeah, so the composition of the blood determines the fate of cells. I go, yes. I said, then what determines the chemical composition of my blood? And they go, the brain. The brain releases chemistry like hormones and emotional factors and growth regulators into the blood, which control our biology. And I go, cool. And then the next step I say is, but which chemistry is released into the blood? And I go, it's based on your mind. What you are seeing and what you are responding to in the world, the mind translates that into chemistry, which is released into the blood, which in turn affects the cells. So I say, so for example, if you're in love and you open up your eyes and see love, your brain releases into your blood chemicals such as dopamine and oxytocin and vasopressin and nerve growth factor. These are chemicals that give enhancement to the cells. So when you're in love, guess what? You're healthier, you're happier, you have more energy. That's because the mind translated the image of love into a chemistry that gives your body health and energy. That's why being in love is so great. And I say, well, wait, what if I open my eyes and I see something that scares me? I go, well, you don't release the oxytocin and dopamine, that stuff. You release cortisol, stress factors, inflammatory agents such as histamine. Basically, you release different chemistry in the blood, so that's changing the growth medium. I go, yeah. And I say, well, the composition of the growth medium determines the genetics and fate of the cells. So it says, when you're in love, you release a growth medium that provides you with health. And when you're in fear, you create a growth medium that actually shuts down your growth and shuts down your system as it conserves energy to prepare for the threat.
This is profound, because what this means is that happiness actually induces chemicals that are connected to it.
Absolutely. How do you know you were happy? You felt it. How did you feel it? Because the chemistry in your body is creating the emotion. But the chemistry that creates the emotion is the same chemistry that regulates the functions of the cells. So it's like, then the way you were experiencing and expressing your life is controlling the expression of your cells. I go, that is the critical point. Because, Kim, look, almost everyone out there was programmed with the same thing that I was programming medical students with when I was teaching in medical school, and that was genes control our lives. And I go, well, if genes control your life, I say, well, did you pick the genes that you have? And you go, well, not that I know. I say, if you don't like the traits, can you change the genes? I go, no. I say, if you got cancer running in your family, are you going to get these so-called genes? I said, well, as far as I know, yeah. And all of a sudden you say, oh, my God, I'm a victim. A victim of what? My heredity. It's these genes that cause me to get sick. It's these genes that cause me to be depressed. It's these genes that cause these problems. And I just receive the genes, and they control me. That's the old belief. That's called genetic control, which simply means control by genes. Then I go, what about the new science? Well, that's the new science that was like the story of the cells in tissue culture. It said those cells were all genetically identical, but the fate wasn't controlled by the genes. It was controlled by the cell's response to the environment. I go, wait, then my cells are not controlled by my genes, but they're controlled by the environment, which is the blood, which is the connection to my mind, what's in it. And I go, well, all of a sudden I say, guess what? You're free to change your mind. You're free to change your perceptions. Well, why is that important? Because it's your mind and perceptions that release the chemistry that control your biology. So as you change your